Okay, hello, this is Susan Becker, and I am sharing my educational presentation regarding a transcription overview for like an undergraduate genetics class. The learning objectives of my presentation, we will define transcription in my presentation. We're going to explain the relationship of transcription to the central dogma of biology, how transcription fits into that um, ideology, I guess you'd call it. We will identify where transcription occurs within the cell, the location actually, where it takes place, and we'll describe the steps involved with transcription, how it begins and what occurs and how it ends. And then finally, we will briefly talk about the role of transcription in gene regulation. It's very important, has a very important purpose in gene regulation. So, transcription is important to understand because it truly is central to biology as a process and as a science. Um, During transcription, the information contained in the DNA sequence is essentially transcribed into RNA so it can later be translated into proteins. So transcription is somewhat can kind of be viewed as almost like the middleman of the central dogma. It's beyond it happens after DNA replication and before translation. So the RNA is kind of the middleman language of the central dogma. Transcriptional control is like I said, a large mechanism of gene regulation. Also, transcription plays a huge role in regulating the expression of genes. And so therefore, it's also a very important aspect to learn. Um, so the central dogma of biology, we hear this repeated a lot in various settings and with various subjects. So that basically refers to like the three legs of the triangle there, that DNA replication produces a carbon copy of DNA from the original DNA template. This is how cells divide by mitosis and reproduce themselves. Transcription is where the information from the DNA sequence is transcribed into RNA, specifically messenger RNA, which is then later translated, which is the third leg of the triangle there, into proteins on the ribosomes in the cell. So transcription occurs in three basic steps. As this is an overview, it's very general. We're not getting into a lot of the details because there are a lot of details that aren't even all that well understood. So it occurs by three steps: initiation, which is obviously where it begins, where the transcription point starts, the elongation step, which is where the RNA transcript is written, and then the termination step, where it ends, where it stops. So there are some differences between transcription that occurs with prokaryotes and eukaryotes, and I'm going to mostly focus on eukaryotes, but I'll throw in a few prokaryotic tidbits here and there too. The prokaryotic transcription process is obviously much simpler than eukaryotes. It occurs in the cytoplasm of the cell versus in the nucleus of the cell, which is where eukaryotic transcription occurs. To initiate transcription, transcription factors recognize the TATA box and bind that. It's a section of DNA where there are repeating thymines and adenines in a certain pattern that is easily recognizable by general transcription factors. And then other transcription factors kind of assemble onto that in our eukaryotic transcription, and a whole complex is formed by the transcription factors assembling along with RNA polymerase. I kind of skipped that part, I'm sorry. RNA polymerase joins after the transcription factors and forms this complex, and that's when transcription can begin. And the transcription factors kind of dissociate from that complex. Actually, in eukaryotic transcription, as the little diagram shows here, there are actually 3 different types of RNA polymerase. There's 1, 2, and 3. They function in forming or transcribing 3 different types of RNA. There's ribosomal RNA, messenger RNA, and transfer RNA, but we're only really going to discuss messenger RNA because the other ones are more functional in translation rather than transcription. So next is the elongation phase, where new nucleotides are added to the strand of RNA. So in this case, the RNA transcript will be almost identical to the non-template or coding strand. So the non-template or coding strand is the other strand that isn't being read directly by the RNA polymerase. And the RNA transcript is identical to this with the exception of uracils being placed where there are thymines in the original DNA strand, because the nucleotides in RNA are uracil, adenine, guanine, and cytosine. Thymine only is present in DNA. The termination of transcription, from what I gathered, is not very well understood in eukaryotes. It appears that it occurs when polymerase actually transcribes a terminator sequence, which can be a polyadenylation signal sequence, poly-A sequence like they call it. In prokaryotes, termination ensues when the RNA polymerase encounters a hairpin loop, which is like a complementary single strand of RNA that kind of double backs on itself and then kicks the RNA polymerase off the chain there. So after the transcription process terminates, the various modifications occur to the RNA to package it and protect it and get it ready for transport. And then as the finished product, the messenger RNA exits the nucleus to be translated into proteins on the ribosomes. So I found a cool video that kind of shows transcription in real time that I would like to share. So I thought that was kind of neat how that little animation showed transcription occurring in real time and kind of summarized what we've talked about in terms of how it happens and the steps in the process. And then this image on the left here is an actual microscope image of transcription taking place. And I also thought that was kind of neat how you could see the actual RNA being produced from the DNA. And I thought that was interesting too, to be able to visualize this actually happening, what's actually occurring. So the role of transcription and gene regulation, like I said, was part of our objectives. It's actually the transcription factors that are responsible for gene regulation. So they bind to the promoters either to turn them on or turn them off. So they would either like allow RNA polymerase to act upon the DNA and perform the transcription or block it. And by doing this, they can control the activity of certain genes. Because if there's no transcription, there's no gene activity, so the genes become suppressed because ultimately there could be no proteins. If there's no RNA, there's no proteins translated, and therefore the gene is essentially silenced. So transcription really does play a central role in gene regulation. There's various research topics that are going on in concerning transcription. Like I said, we understand the basic mechanisms of transcription, but there are a lot of nuances and little finer topics that are not very well understood, like termination and what causes termination, what happens during termination, things like that. As I was researching this, I found that to be a repeating message, that there are certain things that just aren't very well understood. And so I found this one website that listed some topics, current topics that are being researched, and novel mechanisms controlling transcription. How exactly is it turned on and off? Deregulation of transcription during disease. What happens? What diseases affect transcription and how? The role of non-coding RNA and RNA modification of translation. So there are several segments of non-coding RNA that end up being transcribed. What happens to them? What are their implications in the process? It's really interesting stuff. It's very complex and obviously too in-depth to go into for our purposes here, but very interesting topic, very important in the study of genetics and molecular biology and all of the above. And there's my references if you want to follow up with any further investigation. And thank you for listening to my presentation.